In this short video, we'll discuss some common misconceptions that can arise from looking at classic depictions of the central dogma of molecular biology. So let's begin there with its most common depiction. In its most classic form, the central dogma shows DNA pointing to RNA, representing transcription, the process by which DNA is used as a template to produce RNA. Then, there's an arrow from RNA to protein, representing translation, where RNA is decoded to synthesize proteins. You'll also often see a curved arrow from DNA back to itself, indicating DNA replication, where DNA directs the synthesis of a copy of itself. Sometimes an arrow from RNA back to DNA is included, capturing the process of reverse transcription as seen in retroviruses. But while this diagram is conceptually useful, it's also incomplete, and in some ways misleading. One limitation is the implication that all RNA is destined to become protein. In reality, a large portion of RNA molecules transcribed in cells are not translated into protein. These are known as non-protein coding RNAs, or non-coding RNAs for short, and they play essential roles in gene expression, regulation, and cellular function.
For example, transfer RNAs and ribosomal RNAs are critical to the process of translation itself, helping to decode mRNA and build proteins. But transfer RNA and ribosomal RNA are not translated themselves.
Other types of non-coding RNAs include some that regulate the process of translation. For instance, microRNAs or miRNA and Pee-wee interacting RNAs or piRNA, both of which interact with messenger RNAs and usually repress their translation or promote their degradation. Other non-coding RNAs can regulate the abundance or the proper processing of messenger RNAs within cells. These once again include microRNAs and Pee-wee RNAs, as well as short interfering RNAs and small nuclear RNAs, all of which contribute to mRNA processing, splicing, and regulation. Finally, there are non-coding RNAs that regulate the process of transcription itself. For instance, long non-coding RNAs, usually referred to in link RNAs, regulate transcription by controlling the accessibility to DNA within the nucleus. And then there's a new kind of non-coding RNAs that was discovered more recently called enhancer RNAs, which appear to regulate transcription precisely at the level of enhancers. Together, these non-coding RNAs highlight a more complex reality. DNA is transcribed into many different RNA molecules, and most of them are not translated into protein, but instead act to regulate transcription, translation, and mRNA stability. Another oversimplification in the classic view of the central dogma is the idea that a single messenger RNA is translated into a single protein. While that can be true, we now know that many messenger RNAs in both prokaryotic and eukaryotic cells can produce multiple different proteins. This occurs through diverse mechanisms, including the transcription of polycistronic mRNAs, alternative splicing, alternative translation start sites, and RNA editing, all processes that we'll discuss in a bit more detail in class.
So to summarize, the classical central dogma provides a useful framework, but the reality is far more dynamic. DNA does direct the synthesis of messenger RNAs, and messenger RNAs do direct the synthesis of proteins, but that's just the beginning. Most of the RNA in cells is non-coding and serves key regulatory and structural functions. Also, messenger RNAs can yield multiple proteins depending on how they're processed. Finally, and perhaps most importantly, the classic model of the central dogma tends to show a one-way flow of information, from DNA to RNA to protein. But in reality, proteins themselves regulate in every step of this process. Proteins regulate DNA replication by initiating and proofreading DNA replication. Proteins influence transcription by interacting with promoters, enhancers, silencers, and transcription factors. Proteins affect RNA stability and splicing, determining which transcripts get translated, when, and how much. And proteins are essential for the process of translation itself. In short, proteins feed back into the very processes that produce them, forming complex regulatory networks rather than a simple linear chain. Understanding this fuller picture gives us a more faithful representation of molecular biology, one that reflects how gene expression, regulation, and feedback really work in living cells. We hope that you enjoyed this video, and we'll talk to you soon in the next one.